Good afternoon, everybody. Thank you for sticking along. It's really, really wonderful to be here and to look at each and every one of you. Wonderful. You don't need to pronounce my name. You can just call me Nadina, and that's that. I oversee the research activities at n-Lorem, and today I would just like to talk to you why science matters. So what you've heard since the very morning is that you heard that every ASO, every experiment, every data point has really the potential to change and treat a life. But in order to do this, what we really needed to do first at n-Lorem was to really build a discovery engine that was specifically unique for the needs of the nano-rare patients. This was something unprecedented. This is something that we had never done before. Today, I actually really have the privilege to take you behind the scenes of this discovery engine and really show you how we work at the interface of urgency, but also scientific excellence. I would also like to show you how the discovery of an ASO for one patient can actually help in the treatment of 10 others tomorrow. Yes, so that's our mission. So actually, something has happened that is quite unprecedented with the nano-rare community and the guidance of the FDA. For the first time in history, we are actually going from in vitro data to patients without in vivo pharmacology. This essentially means that these in vitro data are going to show us which one of these ASOs is really going to work and which ones we're going to have the first taste of safety. This really means that these in vitro data need to be super duper. They really need to be robust and they need to be QC'd on the most excellent manner. So what we really need to do in my team is deliver individualized ASOs very rapidly, but at the same time very rigorously and safely for further preclinical development and treatment. So we really operate at this interface of speed. The patients, your loved ones, don't really have time to wait. So we really do our best to operate at speed. At the same time, and keeping that balance, you also cannot afford sloppy science. So we really need to operate under this scientific excellence. So this is the first thing that we need to do. The second thing that we need to do is really innovate, advance the technology further to really help even more nano-rare patients today that are not served right this moment. So, of course, the question that arises is, how do we really ensure this balance? How do we do this? How do we both ensure speed and scientific rigor for the most urgent unmet medical needs? And the answer to that is we have built our discovery platform. So we have two very simple but very, very powerful goals at discovery and research. We really need to identify the most optimal ASOs per patient case with really precision and certainty. And now at this moment, I want to say we don't want to deliver for preclinical development any ASO that works. No, we really want to give the ones that we are 100% sure that are a group that work the best. Now, the second thing that we want to do is, as I said, advance the technology. We know that at the moment there are certain diseases or there are certain mechanisms that we are not currently accepted. So we want to advance our technology to treat even more patients faster and even more safely. So these two goals obviously go hand in hand. We want precision for the individual and progress for the community. And this is something really unique that we do at n-Lorem. We don't just make ASOs. We're really building and advancing the science to really ensure the best treatments possible for the future. What makes our platform unique? Well, we have really unmatched experience across millions of ASOs, together with a very scalable, individualized process that really delivers both efficiently and uncompromising scientific rigor. And we really had to build this from the ground up. As I said, this is the first time that this has been done for the nano-rare community. But this platform is essentially is not a standalone. It really stands on 3 miracles of science, and this is what Stan touched on this morning in his presentations: genomics, ASOs, and the power of iPSCs. These advances have come together that actually make possible what we do at n-Lorem today that was not even possible a decade ago. Genomics can actually act as a map, really. So you can really pinpoint, this is really the starting point, we can pinpoint the exact genetic change that causes the disease, and this really gives every single patient a precise starting point to treatment. It also is a starting point for the ASO design, and this is exactly where the treatment begins. The second is really the really powerful tool is our medicines. These are ASOs. We can really create personalized medicines for you and your loved ones that really don't just manage the symptoms, but instead we are really going after the root cause of the patient's disease. And lastly, iPSCs. This is really the testing ground. This is quite amazing, really, because this is the biology of each patient really in a dish. And this is what we are using. We're using the patient cells to essentially discover the best ASOs in a very fast and also very cost-effective manner. Our platform is based on 4 pillars. We are purpose-built for scale, speed, and scientific excellence. The second thing is we have an unparalleled ASO experience. We really have worked on decades on understanding ASO mechanisms, oligonucleotides, and RNA. We really live and breathe in the RNA world, and we absolutely love it. And that essentially means that we have the right experience and expertise to really make the right decision, even if we go through uncharted waters in biology, given these are nano-rare mutations, and these are things that they have never, ever been tested in the lab before, many times. Thirdly, and that was actually something that was touched this morning, there was a question about AI, and I will go through it in more detail later on in my talk. We do, what I like to say, the correct integration of AI. Of course we use AI. AI is also a buzzword at the moment. AI is not, for us, a replacement for science. What AI does and the way we use AI is we feed it our data, our right data. So we actually use it as an accelerator. We use it to accelerate, and we use it to streamline our discovery and research. For example, AI is great at really seeing patterns. If we feed it a lot of data, again, the right data, if we feed it the right data, it can really see patterns that maybe I wouldn't be able to see as fast. However, it is our scientists that really decide which of these patterns are the ones that they are relevant for each discovery case. Fourth is continuous learning. We have a platform, and it's not static. It is essentially continuously evolving. And it's evolving by all of you. It's evolving by the data from you and your loved ones. Every patient is really teaching us something new about biology, about health and disease, and really about ASO mechanisms. And this is exactly what I said before, that this is how we are advancing the science for the treatments of today, but also of tomorrow. Right, scientific rigor. Let's really get to business now. Scientific rigor for us is not a hypothetical. It's really measurable in our outcomes. We have very compressed timelines. As I said, your loved ones and all of you really don't have time to wait, so we do our best to operate at maximum speed. But that said, we hold very high standards for efficacy, potency, and safety. And really, this commitment that we have to you is now really measured in nearly 90% success in ASO discovery in all ASO strategies that we use in discovery right now. Non-allele selective ASOs, allele selective ASOs, and also splicing modulating ASOs. Right, so having all of this knowledge, expertise, and I've given you a little bit of a taste of how our platform works, this actually means that we have really this armor, really, we are very, very well equipped to be able to tackle even the most complex gene targets. And every target is unique, every target is very complex, but I would say I just would like to take as an example here ion channels, which is one of the most complex and also fascinating gene targets. They actually remind us that we are electrical machines. All of you are, I am, everybody. Really, every thought that we make, every heartbeat, every movement, depends really on this beautiful flow of ions, which are essentially charged particles that generate and control all the electrical currents in our cells, in our bodies. This means that the ion channels, these beautiful proteins, are really the gatekeepers. They really finely regulate how ions move in and out, and therefore they're ensuring the proper signaling in the brain, in the heart, in the muscles, and beyond. And of course, you now know that if something is very crucial, usually it's very, very tightly controlled. Cells are very smart. They really want to control and keep balance things that are very crucial. Therefore, this means that even a single nuisance, even a single disruption can actually cause a devastating disease. And this is exactly what happens with ion channels. Essentially, we have a mutation only in one of the copies of the gene, and essentially that can lead to devastating disease. But what happens in ion channels, and as I said, these are really essential proteins, is you still have a copy of the gene that works very, very well. So what that essentially means is that we can use, and you've heard the word, you've heard this fancy word today many, many times, we need to really use allele selective ASOs as the optimal therapeutic strategy. What are allele selective ASOs? Let's take a very brief step back to really show how exactly this works. So each one of you have two copies of your genes. You have inherited one from your mother and one from your father. These sequences, and you can see them there in RNA, in blue, they're very, very similar to each other with some very tiny differences in this sequence, these letters. These differences, we call them, it's a fancy word, single nucleotide polymorphism. These are SNPs, but essentially what these really are just differences in this sequence. Now, if a difference in that sequence, if a SNP is essentially causing the disease and is depicted there in red, we call it pathogenic. If a SNP is not causing disease, we call it non-pathogenic or benign. But essentially, these differences can act as the anchor for us to really differentiate between these two copies. So now you can understand that we use these SNPs to essentially differentiate between the bad copy, the pathogenic mRNA, and the good copy, the healthy one. Now, what comes into play is a really great enzyme that I had talked last year even more about it, which is called RNase H1. So really what we do here for these gapmer allele selective ASOs is we leverage the very unique and very selective cleavage pattern that RNase H1 has. So RNase H1 is very susceptible to RNA sequence and RNA architecture. Even a single mismatch can essentially change how a specific RNA DNA heteroduplex, the RNA together with the ASO, would be cleaved or not. It is this exact thing that we really leverage here, because what we do is we design usually around 20, let's say, ASOs. For each one of these exes is one of these SNPs. And like this, we can then specifically target, and the RNase H1 will recognize only the pathogenic mRNA, leaving the healthy one almost intact. And this is essentially what is let's say, the science behind an allele selective ASO. And this is what we've put into practice. This is really this ASO mechanism that we understand so well, and we really know how to do the design of these allele selective ASOs, that they have given us this unparalleled expertise in allele selective ASOs design and discovery. And essentially you can see here a table, and I can tell you that I'm sure that this table is going to continuously growing. These are all the ion channels that we have in our pipeline and have passed through the discovery portfolio. And all of these are essentially, each one of these are patients that we are currently discovering, or we have finished discovering an ASO for. This really, therefore, our deep experience has enabled us to really target specifically the bad copy of that ion channel, leaving the healthy copy doing what it's supposed to do. So, and I have to say that all of these are toxic gain of function, so this is important because that would be really the optimal therapeutic strategy. Right, now let's dig a little bit deeper. So I told you that what is really, really important is these little differences, right, that we call SNPs. Now, how do we go from identifying these little differences to delivering the optimal ASOs, a group of these optimal ASOs further for preclinical development, again and again and again? How do we do that? And this is where our discovery engine comes in, and I'm very happy to introduce it to you, that essentially stands in three main pillars: scalability, rigor, and decision making. Let's go through them one by one. The very first one is scalability. And how we start every ASO, every case that is accepted, that is going through the discovery engine, we of course start with the ASO design. And there, and this is where your whole genome sequencing that you've been asked to is so important, is because we want to scan for these differences, these SNPs, the entire pre-mRNA. In this case, as I said, we're not just simple ASO makers. We don't want just screen a handful, find some that work, and we're done for the day. We essentially want to find really the best of the best. We don't want to leave any stone unturned. And in order to do that, we really design ASOs almost for every non-pathogenic SNP. We usually screen about 500 or more for some of the ion channels. We have some that we even screen 1,000. And this is where we take them through our primary ASO screen. And this is what we do in patient cells, either fibroblasts, iPSCs, or iPSC-derived neurons. Then from these, we start following it down to about 50 to 75. This is really our scrutinized step. This is our dose response evaluation, where we essentially do a 5.4-fold dilution of the ASO concentration to measure potency, which I'll go through in a moment. And then we also screen about 50 to 75 in the BJAB assay, which is essentially an assay that measures in vitro immune activation. As it was touched, I believe, by several people before, really activating immune activation is a big no-no, especially for the CNS. So we really, really want to avoid these immunogenic ASOs very early on. Then for in vitro assessment, we want the ASOs to be very selective. Therefore, we don't want any off-target effects, and we screen for that as many as necessary. This is essentially, as you can see, it's a funnel approach. So this is all the ASOs that are going through our funnel. And essentially, we want to shortlist the most optimal ASOs of this 500 or 1,000. And these are essentially the top 20 optimal ASOs that we think are the most efficacious, potent, allele selective, we're talking about an allele selective approach, and safe, that we then hand over for tolerability studies and preclinical development. Right, okay. Maybe it got tired, let's see.
Oh, the battery is low.
Should I come?
It's been a long day.
Yeah. Should I come here and just,
Yeah.
Yeah, that's fine. No, that's fine. Okay. let me just make sure that this, okay, great. All right, then I'll see more of you here. Okay, so, all right, so scalability, we talked about scalability, scientific rigor. So if scalability is really designed to cast a very wide net, as I said, we really do not want to leave any stone unturned. Scientific rigor is there to really, really ensure that we have the most optimal ASOs that are going through each and every one of these stages of the funnel. For example, for the ASO design, and these are, oh, fantastic. Okay, I'm back on. So for the ASO design, and this is essentially work that we do through our AI-driven algorithm. I will talk about this in a minute. We have all the important motifs that we know that work from our past experience included, and all the ones that we know that are not good, essentially excluded. For the primary ASO screen, nothing passes through unless we have an 80% of the target reduction. This is essentially our efficacy for the target engagement. For the dose response evaluation, this is really, the primary screen is really at the highest ASO concentration. The DRC, the dose response, is done, as I said, in a 5.4-fold dilution. And this is how we measure our IC50. And this is how we measure our potency for both the mutant and the wild type. And we obviously want the ASO to be very, very potent. And there, we also want to measure our allele selectivity, which is the IC50 of the mutant versus the wild type. And there, of course, we want it to be at least 10-fold selective. On the B jab, we really want at least nothing will pass through that is more than 2-fold of increase in a TNF-alpha surrogate, which is called CCL22. And then for our off-target analysis, as I said, we don't want any unwanted effects. We want the ASOs to be very selective. And there we want at least a 10-fold difference in the ASOs for target RNA versus any off-targets. And this is how we get to select really these top 20 most optimal ASOs. Now, scalability and scientific rigor don't really have a lot of impact if we don't make the right decisions. And as I said, our decision making is very, very important and is really at every single step of this procedure that essentially we have a go, no-go step at every of these steps to really ensure, again, our goal is to have the most optimal ASOs that are going through to end up with this top 20. Some of them, there are some questions that I'll give some examples. The very first is the whole genome sequencing, the data that we really need that are sufficient for our ASO design? Or for our primary ASO screen, do the patient cells meet really the QC requirements? Or when we go through dose response evaluation, do we have the optimal allele selectivity, potency, and efficacy? If the answer is no, what we do is we go and rescreen. We go and redesign ASOs in some cases, and we pass new ASOs through this funnel. Decision making is very important. This is essentially how we funnel, and we make sure that we have the really top, most optimal ASOs possible. If that is not possible at this stage, we go back and redesign and rescreen if that is at all possible. And here, this is again an example of the ion channels. This is how our screens look like. And this is on the top panel. This is essentially the primary screen, which means that this is a portion, and this is CACNA1A. This is a real-life example of a calcium ion channel. And essentially, what you will see here is this is a portion of these ASOs that they haven't been administered in the patient cells. This is the highest ASO concentration. And obviously, what you are expecting to see, what you want to be seeing, is you want to be targeting this, given that this is an allele selective approach, you want to only be targeting the mutant mRNA while leaving intact as much as possible the wild type. This means that you want the red to be reduced as much as possible and the blue to remain intact. And you can already start seeing that there are some clusters. This is a plate 1 of 20, right? You can already see a cluster of some of these ASOs already emerging. And then on the bottom panel, this is an example of dose response screen. This is an example of DRC. And you can really see what you want to be seeing there. You want to see a dose-dependent reduction of your red, which is your mutant. This is the bad copy. This is a bad RNA. And you want to see an either flat blue bars, or at least not as dose dependent as the ones in red. And obviously these are the ones that we use to calculate the IC50 and determine how potent these ASOs are. This is a real life example. These are the top 20 candidates that they had been brought forward for tolerability studies and preclinical development for the CACNA1 ASOs. And luckily, actually, these ASOs could be used for a second patient. I'll be going for that in a moment. Now, our discovery engine works because it really is built on very, very strong foundation, which I talked about at the beginning. This is not really a trial and error situation. It's not just miraculously working. We know exactly why it's working, because it's the correct one. This essentially means that we can take through our discovery platform a lot of different gene classes and therapeutic areas. And there we have a very broad therapeutic reach, because we can see the same rigorous process being applied to neurological, hepatic, and other disease areas. And you can really see here different targets that we have been tackling, both ocular, which Shyamanga talked a little while ago, but also hepatic or kidney, but also, of course, ALS diseases and neurodevelopmental. And this, of course, have been through different routes of administration. Now, all that I've told you today essentially wouldn't really mean anything if we were not having positive clinical data, and if we were not really making an impact in our patients, which, as you heard today from Laurie at the beginning of the day, we are. So we now have a fully automated high-capacity ASO screening capabilities, and I have to say that this step in the whole discovery and development is really the most rapid and cost-effective. I'm very proud to say for the team that we have a very, very high productivity rate. We can clear about 8 to 9 patient cases per quarter. This essentially, just to give you into perspective, it means that we can have more than 1 IND per person per year, and this is definitely much more productive than any organization or drug modality that we really know of, which means that we have fully scalable systems and laboratory facilities sufficient to support more INDs and treat more patients tomorrow. As we said, to link it to the previous panel, the only impediment is the funding. And on the right corner of this slide, essentially, you can see the machines, and these are the robots that ensure that we are doing everything in a very high throughput manner. So these are really the robots that allow us this fast and error-free turnaround. But these on top are essentially the people. These are the people that are really behind the machines and come in every day, turning science into treatments very fast. So the very first part of my talk, what I showed you was I showed you through our discovery engine how we deliver the most optimal ASOs further for development and for treatment. This is our discovery engine, which works. But we don't stop there. We're not satisfied with just that. We're not just executing a process. We're looking ahead and we're looking into the future. We essentially want to enable innovation and be able to treat more patients today than we could treat, more patients tomorrow than we can treat today. And this is exactly where the second part of my talk is essentially focused on. I will show you where science becomes solutions. And this is a little bit of what was touched early on. This is about the proprietary AI-driven ASO design platform. And this has, of course, been built from 35 plus years of foundational ASO expertise and really millions of ASOs. It is really continuously refined. We are continuously learning from our own very high quality data that I just showed you of how we make them. We really have a multi-omics integration and we also integrate all our in-house and published data sets. Now, the important thing is that we don't just want to stop at just the design platform that we've already established in-house. What we really want to do is, even for allele-selective ASOs, really try and find patients that they can be served under the same ASO. Obviously, this can mean that we can treat patients faster and much, much cheaper. So what we have done is we have the internal capabilities to really turn this discovery engine, our ASO design platform, to really identify and build these SNP ASO maps to really allow this rapid identification of the patients in the pipeline. And actually, we have done this for multiple targets now already. We have done it for KEF1A, we have done it for SBTLC1, we have done it for DHDDS, CACNA1A, and also TARBP. And I would like to show TARBP really as the poster child here, that we have managed to really do this. And of course, this is particularly important because, as it has been touched on before, ALS is obviously, and I don't need to spend a lot of time talking about this, ALS is a devastating disease, full stop. The patients of ALS really don't have time. We don't have time, any time to waste. So what our goal here is in research is to essentially identify if we can have ASOs already that we have discovered that several patients can be benefit for. But in order to be able to do that, we need to really do the hard work of having all the SNPs, both non-pathogenic and pathogenic, mapped. On the left-hand side, you can see all the patients that we have accepted in our discovery portfolio. And here you can see in blue the TARBP. It's a relatively short gene, which actually makes it easier in a way to actually do that. And here you can see all the SNPs that we have mapped based on the patients across the gene. The gray depict the non-pathogenic SNP, and the red depict the pathogenic SNP. As I said at the beginning, what we usually do for the design is we normally design around 20 ASOs around each SNP. So we now have all of this knowledge, knowing where our ASOs are in correspondence with each non-pathogenic SNP for each of the patients. This knowledge is power, literally. Because what we have been very successfully been able to do is that we have been able to identify specific leads that we have essentially discovered that they can be shared and used by different non-related TARBP patients. And you can see at the bottom really the number of compatible patients that you have with each of these lead ASOs. And the numbers are astonishing. If you think that this is actually really, it was designed to be individualized and it was supposed to be for one patient. And we have 5 that are served by that lead ASO, 3 and 2. And the very important thing is we also have some and where we have managed to discover an ASO that is targeting the pathogenic mutation. This is the second example. The first example was shared by Xiaomanga just a little while ago, where we did the same for the ocular target. This is obviously very powerful because it means that all the ASOs that we have funneled down, since they can target the pathogenic mutation, any patient with this mutation could potentially benefit. So this is one of the ways that we're really tackling all of these frontier problems in ASO discovery by really advancing our technology to really reach more patients in a safer, better way. And some of the areas that we are really focused on, and these are what we may call next generation ASO solutions. The very first one is allele selectivity, and this is a big one because more than 50% of our patients that we accept require allele selectivity. So really being able to refine that But even going back to patients that at the moment we have declined because of the lack of allele selectivity is very important. The second that comes directly after that is obviously design ASOs for RNAs that they're very, very small, structured, and very heavily modified. The third, and this is for people who are asking, this is all the loss of function mutations. At the moment, what we can do with ASOs is either, of course, get rid of something that is causing a problem, and this is our gapmer ASOs, or modulating splicing, and this is our splicing modulating ASOs. At the moment, we cannot essentially create ASOs to increase more of protein levels of something that is missing by a loss of function mutation. We are really now and have a lot of advances that we expand to treat many of the patients that we currently have to decline. We also minimize innate immune activation. We already have safe ASOs. We want to make them even safer. And we also want to understand the molecular and cellular pathology of the nano-rare mutations. One of the problems of the nano-rare mutations, of course, is that they are not really reported. However, for us at research to be able to say this is the ASO strategy that we will follow, we really need to be able to understand what this mutation does. So sometimes we need to really take a step back and understand this molecular and cellular pathology before we can actually begin drug discovery. And finally, there is a lot of things out there in the literature. Not everything is right. So when data don't add up, we really dig deeper and we ensure that really is the science that drives the solution and not assumptions. And I will take you through some of these examples. I will be as high level as possible. A lot of members of my team are actually just there standing and they have posters that they will give you more details on. But I just want to give you a flavor of all the research work that we essentially doing to enable more treatments. So the case study of GIP3, that was a patient that came to us, which was essentially we didn't really know if this mutation was a toxic gain of function or a loss of function, meaning we didn't know if actually ASOs would be an optimal therapeutic strategy, and if so, what would that be? Would that be allele selective? So GIP3 is a very important protein. This is a scaffold protein that essentially is very important for neuronal function, and it's important to really transfer cargo along the neuronal axons. So through going through a series of experiments, we were able to identify and establish that this mutation is indeed a toxic gain of function. Among other cellular and molecular events that we really unraveled, including this mutation was disrupting axonal transport and causes apoptosis and cell death and all of that, we were able to actually uncover something really novel and unique, which is that it disrupts dopamine D1 signaling. And that was very important because actually for the pathophysiology of that specific patient, what we can also say is that perhaps this is a new cause of juvenile Parkinsonism, which means that maybe Parkinsonism or part of it can actually be caused by GIP3 mutation. So really what we essentially established was that the allele, that since this is a toxic gain of function, an allele selective ASOs is really the optimal therapeutic strategy, and therefore we could really, with the ASOs, we were able to rescue each one of these phenotypes. And if you want to know more, Wei has all the details on his poster and is also published. The second example is a case where actually that specific mutation was published. Now, GB1 is a really crucial enzyme that essentially when it's important for glycogen biosynthesis, and when it's mutated causes APPD. And one of the second most common mutations in GB1 is essentially an insertion deletion, an indel. Now, even though that was published, there was a lot of ambiguity about which was actually the sequence of the indel. Also, we knew that this mutation causes a splicing defect. However, we didn't really know the nature of exactly what was happening. Now, these are not minor details. These are really fundamental questions that we need to have answered before we actually start an ASO design and discovery. You need to know the exact sequence in order to be able to design an ASO that is going to work. Or you need to know what this mutant transcript is doing so that then when we test with an ASO, we are able to see that it rescues. To cut a long story short, we really took a step back and we decided to perform our own research and really just repeat some of these experiments ourselves and perform the sequences and really find out. And we were really successful with that, and essentially what we managed to do was to essentially, of course, rescue the phenotype, find the sequence, and now we have these ASOs, and we are in the development stage for this program. Right, we are already delivering, and this is really, there was a topic of this morning session, we are already delivering safe ASOs. We want to make them even safer. We're not even satisfied with that. Now, the risk is that phosphorothioate ASOs can really trigger the innate immune activation through TLR9. And this is something that you really do not want to have, especially in the CNS. And this response is quite unpredictable. ASOs can be agonist, can be partial agonist, or not induce activation. The risk of that is obviously toxicity, which is something that we do not want to have, is burden of screening. We screen just endlessly, just in case that we have immunogenic ASOs that need to be wiped out, which means that we have a loss of therapeutic opportunity. Now, in order to tackle and solve a problem, you obviously need to understand the problem first. So our goal was to determine how PSASOs really terminate the innate immune response. As I said at the beginning, cells are actually very, very smart. So we thought, if we actually find the endogenous mechanisms, really the endogenous metabolites that really are terminators, what we can do is we can use these as excipients in our ASO formulations of intrathecal or other administration. And Julia can certainly talk to you more about that, but essentially what we found was with these two very important enzymes and the tryptophan oxidation products were the ones that they were responsible. So it is possible, but that by using them as excipients, we could indeed make safer ASOs for intrathecal and peripheral administration. Allele selectivity is very complex. It can be very cumbersome. At the moment, what we're doing is seeing the SNPs, design the ASOs, continuously. It's like that's a process. Wouldn't be much better if actually we could find a smarter way and more precise way that we could really refine this allele selectivity, and even for more complex targets. And this is exactly what we're doing. And we are leaders in allele selectivity because really we have a deep knowledge of RNA and RNA stage 1 enzymology. And we can really exploit the unique cleavage patterns for true selectivity that come down to the RNA sequence and RNA architecture. So far, we're taking advantage of the RNA sequence. It's now the RNA architecture that we also want to integrate through genome-wide databases. Also, we're doing novel chemistries to really minimize immunogenicity and immune activation. And I want to use one case study for this, for really refining the allele selectivity, which is the RNU42. We had a question earlier on. This is the Renew syndrome. These are essentially mutations in U-rich small RNAs, and these are very important for a very fundamental procedure, which is called splicing. And this is a really, really rapidly exploding field. This happened, the diagnosis happened a year ago, and that has been insane. Renew currently appears to be the most prevalent and best characterized of all these U-rich RNA NDDs. And what we're doing when we accepted our patient, first it was great, we're RNA people, we have an RNA disease. Whew. Problem is, the thing we didn't really know, as with GIP3, if this mutation is indeed toxic gain of function. So we really needed to get to work and really define what is the best ASO strategy. Do we have an ASO strategy? We now know that indeed this mutation is toxic gain of function. The other thing, this is a tiny, tiny RNA. This is 142 nucleotides long and very, very heavily structured, one SNP. So how do we actually refine allele selectivity? Well, we have gone to work, and now we have some very promising ASO candidates for this patient, and we'll be able to give you even more information. And last, and as I start wrapping up here, we really want to treat more patients. And at the moment, the one that there is a huge unmet medical need is obviously for loss of function mutations and haploinsufficiencies. 40% of the drugs are agonists. As I said, most of them we are reducing a bad copy, but for many diseases, we need the exact opposite. We really need to upregulate most of that missing protein. And what we're doing is, the great news is we have a lot of foundational work from all the lab that Stan did in his lab over at Ionis, so we have a lot of proof of concept established. Now we take this foundational work and we make it an even step further, and now this is where we want to start really pioneering and unlocking these treatment possibilities for these patients. And Anthony will be the one that can be showing you more details, and we don't have one strategy right now that we are exploring. We have around three more, but I think the most well-developed at the moment is yours. And if you were to just very quickly, and this is what I just want you to think, is that normally what you would have in a haploinsufficiencies is you would have a copy that is mutated, causes loss of function. The wild type is still there doing its job, but usually that's not enough. So you want to upregulate that to be able to restore that function. Now yours that they are upstream at the 5' end can actually act as a decoy in translation, causing either dampening of translation or no translation at all. So one way would be for us to use ASOs to block these URFs to boost gene expression. And as I said, can learn more of the details later. So if I were to just summarize the second part, I would say that we are really expanding the boundaries of ASO technology today. And what we're doing right now, we have 90% of discovery success, and we are delivering potent, efficacious, and safe ASOs. We definitely can treat more patients today than we could yesterday. And our next step is obviously improving allele selectivity, expanding upregulation, and minimizing immunogenicity. But we're not even stopping there. We want to go beyond and really expand our therapeutic reach. So at n-Lorem, the next chapter in ASO advancement is not somewhere just in the future arbitrary, is really already underway. Every challenge that we are unlocking with our discovery cases can lead the breakthrough of the future. And really, this is this complete circular motion, because the unique thing is every patient gives us data. The data are really help us to gain our knowledge. The knowledge is power, is really the catalyst that makes us think even better solution for the patient that comes next. This really feeds into that circular motion, and then something extraordinary happens. We go actually from patient to hope, because then we can have the tools to treat even more patients. I think I've talked all of that. We are really creating the knowledge. We are publishing a lot. You will see a lot of this work in the posters. We're filing patents, and we are really, the most important thing is with the efficiency and quality here are really, oops, how do I go back? Oh, it's okay, I can say it. If I were to leave you with just one single thing, I would just say that we are really built for urgency. This is what our discovery and research engine is really doing. We are really hear you, and we want to deliver as fast as humanly possible. And we are really built for hope. So this is what we are trying to really advance the technology to be able to address the more and more needs of the nano-rare patients. And while leaving you, I would just, none of everything that I've told you today would have been possible without my fantastic team here, that they're just coming in and believing in science that it can really deliver for you. And with that, thank you very much for your trust and for inspiring us. Thank you.